In this lecture short, I'm going to talk about the different properties of membranes. We're going to look at the fluidity, selective permeability, potential, membrane potential, its ability to self-seal, leaflet assembly, and then lipid rafts, which are these microdomains of unique functions. Okay, so first we can talk about the membrane fluidity. So remember, in the fluid mosaic model, all the lipids are very freely moving. So fluidity is referring to how viscous the different lipid bilayers are. And the one thing is that cholesterol is going to add rigidity to the membrane. So this is a really fun, very insightful cartoon of with the membranes. So you've got different types of phospholipids here, and it's showing you those alcohols. So oxygen's red, and there's our phosphates, but here's those free fatty acids. So those long hydrophobic fatty acid tails, and those are a lot longer than your typical cholesterol molecule here. And then don't forget, the cholesterol has that 4-fused ring system, which does not freely rotate whatsoever. That is a very rigid part of the molecule. Many of your free fatty acids have lots of single bonds, and carbons around single bonds are always freely rotating. So those are just like wet spaghetti noodles, like flipping and flopping and twisting around. But again, the fused ring system of the cholesterol molecule is very rigid. So you cram in more cholesterols into the membrane, right? And it's going to make it very stiff in pretty much the region where the distance of how long that 4-fused ring system is. And then you get a very pliant or very less viscous part towards the middle, which is where just those longer fatty acid tails can kind of be wiggling freely. So you get a lot of movement there. But so the more cholesterol you add, the more rigid and stable and stiff your membrane becomes. So you can add too much cholesterol. That could be a thing. You don't want very stiff, rigid membranes, right? You want them to be fluid to some extent. Okay, membranes are selectively permeable. What does that mean? Well, when you think about molecules that have to pass both this polar and this nonpolar region, the only thing that can freely diffuse across the membrane is very small nonpolar compounds. So things, gases, the big ones in biochem is carbon dioxide, because many of our metabolic processes generate CO2 as a waste product. And then that just freely, it's a gas molecule, it just freely diffuses to areas of lower concentration. And they don't have to worry about being exported through a protein or anything. They just can diffuse completely across this hydrophobic region. It's the same thing with oxygen. So we actually take in our oxygen, right, to drive a lot of our metabolic processes to generate our energy or ATP. And oxygen is a very small nonpolar molecule as well. So we breathe in oxygen, it's getting carried around on our hemoglobins, and then it gets released in areas where the oxygen concentration is lower, and then diffuses, down its concentration gradient. And then usually that's to the areas where we need, like the mitochondria are consuming massive amounts of oxygen to generate ATP for us. Things that will not cross the membrane, anything charged. No, not going to happen. You're never going to get a charged molecule through all that hydrophobic space. No way. And then bigger polar molecules. Even I think water doesn't easily move into this hydrophobic region. We actually have lots of particular proteins called aquaporins I'm going to talk about that help water move. But again, water doesn't easily just like fly across this membrane. This is very hydrophobic. There. Okay, so having a membrane allows for a membrane potential. If you've taken any neuroscience classes, you'll know that the average potential across any membrane is about -70 millivolts. We have a highly positive charge on the outside of the membrane and more negative charge on the inside of the membrane. Typically, that's due to like phosphates. This A- is like phosphates, carboxylates, some calcium, some chloride. Those are going to be your negative charges. And then on the outside, you have lots of sodium on the outside of the cell, lots of, not a lot of potassium really, but you just have more sodium than chloride there. So that's going to give you the overall net positive charge. But so we have membrane, having the membrane can allow us to have a membrane potential, which is how neurons function. And without neurons functioning, definitely eukaryotes, multicellular eukaryotic beings like us humans would have a really hard time doing anything. Like the nervous system does everything we do, breathing, talking, thinking, right? So, okay. Okay. Membranes also have the ability to self-seal. So let's say there is a disruption, like you get cut, and that's a major disruption. And what happens is you break the membrane. So now there's this exposure to water because again, water's everywhere, right? We're swimming in water in our bodies. So what'll happen is there is an entropy-driven self-sealing. So notice that right now, for a very, very, very small fraction of time, those hydrophobic regions are being exposed to water. And then think of like what I said in biochemistry class of how now all those water molecules are very ordered around the hydrophobic tails, and that is decreasing our entropy, and that's not favorable. So usually what you'll see is if there's a hole in the membrane is that all of the polar head groups, it kind of like caves in on itself. So the polar head groups are exposed to the water part, and that's going to free up water molecules that were ordered, thereby increasing the entropy, which is why it's entropy-driven sealing. And then eventually you can reassemble and fix that bilayer. And that's just kind of the slides from class talking about how this is a positive entropically driven, how, again, we're freeing up a lot of ordered water molecules when we aggregate the hydrophobic regions together. Okay, the composition of the outer and inner leaflets are asymmetrical. I mentioned this before in an earlier lecture, but here's some data to show you that. So we're looking at 3 different types. This is a bar graph. I really like this bar graph, actually. It's 3 different types of cells. You've got a human red blood cell, a rat liver cell, sorry, something just popped up, and then a pig platelet cell. Sorry. Okay. And so what we're looking at is PC is the phosphatidylcholine. We've got phosphatidylethanolamine. We've got phosphatidylserine. These are 3 different types of phospholipids. And then SP is the sphingomyelin. So this is our sphingolipid here. Oops, sorry. Go back. There it is. Okay, so in the human red blood cell, you can see that there's a very high percentage of sphingolipids on the outer leaflet and not so much on the inner leaflet. Like phosphatidylcholine and sphingolipids are on the outer leaflet, and the major components of the inner leaflet are phosphatidylethanolamine and phosphatidylserine, right? But so I just like this bar graph because, again, the greens are showing the percentage of things on the outer leaflet, and then the orange is showing the inner leaflet, and they're not the same. So again, flipases, flopases, and scramblases are helping to drive that asymmetry. Okay, I think this is lastly, I wanted to talk about microdomains, which are called lipid rafts. These are dense regions of specialized proteins. And they're called lipid rafts. So this image, which looks really, really like falsely made, but it's not, was an atomic force microscopy image, and it's showing some of the lipid rafts. So atomic force microscopy is this type of microscopy where if you have your sample and there's this tiny teensy needle, and the needle is essentially dragged across the sample, and it's a very sensitive needle. So every little bump, the needle pops up like to go over a bump, and it measures that. So you're just measuring the surface, like what's on the surface, like the topography of whatever it is you're looking at. It's similar that if you were to take a penny and put a piece of paper over it and then take a pencil and kind of like, sketch a pencil, how you could see the shape of the penny and the outline of the face of Abraham Lincoln and all that. Because that's like that's the demo for how atomic force microscopy works. Okay, but you can see that on this membrane, right? The needle's coming across, and then boom, it hits this like one region, and then it comes back down. So the down parts are going to be your lipid membranes, those lipids, the lipid molecules. But these dense regions, you're going to have lipids there too, but probably very highly concentrated regions of proteins. These are going to have your signaling, your receptors for your signaling molecules, right? These specialized little dense raft regions that float around all together are used for endocytosis, exocytosis, and signal transduction. Okay, so again, these are called lipid rafts. Okay, so what are the takeaways from this mini lecture? Hopefully, at the end of this lecture, you could define in detail the following properties of cell membranes. You could talk about the fluidity and how cholesterol affects the fluidity, but also why cholesterol affects the fluidity of the membrane. How membranes are selectively permeable and what types of compounds can pass the membrane and which kinds cannot pass through the membrane. Talk a little bit about membrane potential, leaflet asymmetry, lipid rafts being microdomains, and that the ability for a disrupted membrane to self-seal is entropically driven.